Hello, my name is Minghui Li. Today I will talk about a branched DNA assay for HIV. The goal of today's lecture is that students will be able to properly perform the branched DNA assay for HIV. After today's lecture, students will be expected to be able to explain the principle of branched DNA assay, discuss the advantages of branched DNA assay, and perform the branched DNA assay for HIV with 100% accuracy when provided with a procedure manual. Before I talk about the branched DNA assay for HIV, I want to first test how much you know about clinical laboratory tests for HIV. The first question is, which test is the screening test of HIV infection? The correct answer is the ELISA test. ELISA test mainly detect HIV antibodies, and they can detect antibodies as early as 3 weeks after exposure. The ELISA test for HIV is highly sensitive, but this test has relatively low specificity. It means it may produce false positive results. The second question is, which test is the confirmation test of HIV infection? The correct answer is Western blot. The Western blot is the most common test used to confirm positive results from an ELISA HIV test. It has high specificity. The last question is, what test can be used to detect HIV viral load? This question is a short answer question. The correct answer is nucleic acid test. Testing HIV viral load can be used to monitor HIV progress and treatment efficiency. Nucleic acid test can also be used to confirm HIV infection, while the Western blot result is not clear. Now let's talk about branched DNA assay. Branched DNA assay was developed by the Kirin company in the early 1990s. Branched DNA assay is one of nucleic acid assay. This assay can be used to detect DNA or RNA. This assay is a signal amplification assay, not a target amplification assay. In this assay, the DNA or RNA will not be amplified. In other words, the number of DNA or RNA will not be increased. Branched DNA assay currently is mainly used for viral load quantification for HIV and HCV. In the procedure of branched DNA assay, the first step is the release of DNA or RNA. The next step is the complementary hybridization of target DNA or RNA with capture extender molecules and label extender molecules. Then the developed big molecules containing target DNA or RNA are fixed to microwell by complementary hybridization with capture probes on microwell. After the target DNA or RNA are fixed to microwell, preamplifier molecules will be added into microwell, and they will complementarily hybridize with label extender molecules. Amplifier molecules will be added next, and complementarily hybridize with preamplifier molecules. After the hybridization of the amplifier molecules with pre-amplifier molecules, a large amount of alkaline phosphatase modified label probes will be added to complementarily hybridize with the amplifier molecules. In branched DNA assay, the adding of series of probes results in the formation of a large hybridization complex on the target DNA or RNA, which can contain more than 10,000 alkaline phosphatase molecules. The last step is adding alkaline phosphatase substrate into microwell, and the produced chemiluminescence will be detected and analyzed by a branched DNA analyzer. The intensity of chemiluminescence is proportional to the amount of target DNA or RNA present in the sample. Currently, branched DNA assay is mainly used for viral load quantification of HIV and HCV. If different concentrations of recombinant HIV RNA are tested for the intensity of chemiluminescence, a standard curve can be made. If the intensity of chemiluminescence is known about the tested sample, the concentration of HIV RNA in the tested sample can be got from the standard curve. Branched DNA assay has several advantages. The first advantage is this assay is less labor-consuming because it has a simple sample preparation and no amplification step. It's also highly amenable to total automation. The second advantage is this assay is not as susceptible to contamination as PCR because it's a signal amplification method, not a target amplification method, like PCR that will replicate contaminated DNA or RNA. The third advantage is this assay has no need for enzyme like Taq polymerase, which means no false negative results due to potential inhibitors to enzymes. The fourth advantage is this assay has a very high sensitivity. The limit of detection of branched DNA assay is 50 copies of HIV RNA in 1 ml plasma. The last advantage is this assay can be used to test viral load. To make sure that you really understand the branched DNA assay, let's do a test with a few questions. The first question is, branched DNA assay can be used to detect The correct answer is DNA or RNA. The second question is, branched DNA assay is what kind of assay? The correct answer is signal amplification assay. Branched DNA assay is a signal amplification assay. It's not a target amplification assay because in this assay, the DNA or RNA will not be amplified. The last question is a true or false question. The question is, false positive caused by cross-contamination between samples due to excessive amplification or carryover is less likely in branched DNA assay. The correct answer is true, because branched DNA assay is signal amplification assay, not a target amplification assay. I hope that you answered all these questions correctly. That's all for today. Does anyone have a question? If you have any question about today's topic, please contact me by email or visiting my office. Thank you.